All right, so thanks so much for giving me the opportunity to present our work on heart slices today. So the story starts from here. So far, the first time any drug touch a human heart is in clinical trial. So all of the preclinical studies that's been done before any clinical trial has been done either on animals or in silico or in vitro. So basically this led to a major issue that there are many drugs, even after marketing, they got retracted due to cardiotoxicity. And cardiotoxicity and liver toxicity, of course, are the top two toxicities that's for marketed drugs. So what we wanted to do is to make a simple, reproducible, and medium throughput culture system to fully preserve human heart slice viability and functionality for at least 6 days. So this could be a model to test at least for cardiotoxicity. And you can as well test efficacy for new cardiac therapeutics as well. But for the toxicity, you have to test for cardiotoxicity for any drug. It's not just for heart-targeted drugs. So what we've done is we have established a system where we get human hearts or big hearts. You can do this on anything from rat and above in size. You can't do it in mouse because the mouse is too thin, but you can do it on rat, big cat, dog, whatever you want. And you can do it in humans as well. So we cut the human heart into blocks. As you can see here, we use a vibrotome to cut thin slices. It's 300 micron slices. And then we culture them under optimized media, electrical stimulation, and we provide oxygenation as well to the media. And this keeps them for 6 days in culture. And we published this in 2019 in Circ Research. And we have a Jovi paper as well if you want to go through the full protocol. So we want to really spread this as much as we can. We helped people to establish this technology in their labs. We are working with Anabars as well. We got their team to be trained in our lab to transfer the technology. So the technology is so easy, it doesn't cost too much. It costs about $30,000 exactly. You need to buy a vibrotome and a CPS system to do the electrical stimulation, and that's it. And everything else is easy. So, and we are happy to help anybody to establish this in his lab. So what can you do with this? You can do a lot of readouts. It's a heart tissue, so you can do calcium imaging. You can isolate adult cardiomyocytes from them and do electrophysiology or do calcium on them or do contractile function. You can do force measurement on the tissue itself, so you can hang it in force transducer and measure the contractility. You could do metabolic flux, you could do transcriptomics, electron microscopy, immunofluorescence. You could do spatial transcriptomics as well if you want to. And we collaborated with Tim McKenzie as well to isolate single myofibril from these heart tissues to be able to even see how the twitch force is affected in just single myofibrils. So this is basically the take-home message. I'm gonna now tell the story of this marathon and how it started and where we are now. I think we are probably in mile 5 out of the 26 miles of the marathon, but we're still too early in this. So why did we get into this? I'm not really a heart slice person. I'm not a cardiotoxicity person. I'm nowhere near any of this. But I was at the Gladstone Institute, and we started a company called Tenaya Therapeutics, which is now based in South San Francisco. And they IPO'd a year ago, and they now have 3 sites. But in the start, Tenaya Therapeutics started on my bench, so I was the first scientist in Tenaya Therapeutics. And we were developing gene therapies to induce cardiomyocyte regeneration using either direct cardiac reprogramming or induction of cell cycle. into cardiomyocytes. And I didn't want anything to fail, basically. And one of the major reasons that many of the therapeutics, of the cardiac therapeutics failing, because they haven't never been tested in human heart before they go to clinical trial. So that's why I wanted to establish this technology. And eventually I established this technology just to do this figure eventually. This was one of the figures in the Circ research paper that we published, the health slices. And to prove that when we induce the cell cycle in cardiomyocytes using these 4 cell cycle factors, we can induce the phosphorestone H3, which is a G2M phase, and we can get increase in the twitch force in these heart slices. And basically, this was the whole reason for it. And this was the start of the marathon eventually. But when we published this paper in 2019, I got a phone call from Alex Ribeiro in the FDA. He was one of my colleagues in the Gladstone. He moved to the FDA. And then he told me, Tamer, we've been dying for something to test cardiotoxicity on. And the IPS doesn't really tell us what it should tell us. So why don't you try this model for cardiotoxicity testing? So then I went to the FDA. I gave a talk there. I talked to many people there. And then we started a collaboration with Alex, and we started to collaborate with Igor Efimov, and I brought in as well a cardio-oncology person, because I'm not cardiotoxicity person anyway. I know nothing about cardiotoxicity. So I got Javed Moslehi, who's an expert in cardio-oncology, who's now in the UCSF. And we've started to do some testing and some validation for this heart slice, platform to see whether it will really tell us the true cardiotoxicity that we're expecting or not. So this is just a figure where, these are different non-cardiotoxins. These are drugs that's been in the market. And they have cardiotoxic effect. And we know that they have, but they're still in the market because they treat cancer. So we are dealing with this. So we choose 3 of them. So we choose doxorubicin, trastuzumab, or the HER2 antibodies, and the sunitinib. So from the FDA, this was the work from the FDA. And from the FDA, the doxorubicin and Herceptin, they have cardiotoxic effect. But the sunitinib itself, it doesn't show cardiotoxicity until you go over 10 micromolar. And this 10 micromolar is really not the clinically relevant dose. The clinically relevant dose is around 0.5 or 1 micromolar. And at these dosages, they actually don't show any cardiotoxicity. So this is basically an apoptosis assay on human iPS cardiomyocytes. So the human iPS cardiomyocyte doesn't really show that much of an effect. So we started to test these drugs on heart slices. And what we did, we took the doxorubicin, Herceptin, and sunitinib, and we did 3 dosages. The first one is the Cmax, and then we did 10 times of the Cmax after this. And we assessed calcium transients, we assessed connexin 43, and we did transcriptomics to see even the mechanism, whether they will show us the actual mechanism that we are expecting, that we see in the humans or not. So if you look here for the connexin 43 is a gap junction protein and the troponin is the cardiomyocyte staining, the cardiomyocyte protein as well. So you can see the cardiomyocyte, the connexin, and in the control slice, you can see the connexin is in the gap junction, it's localized correctly, and so on. With the doxorubicin, you get really a big disruption in the connexin. With trastuzumab, basically you are killing a lot of cardiomyocytes as well. You're even getting the troponin out. But with the sunitinib, you don't really get that much structural defect. So the connexin and the troponin are really still good. So what about the functional effect? So this is a calcium transient in control slice. So you see that the slice is contracting, and the calcium transients are really good in there. I need to play this one. Okay, here you go. All right. So for the doxorubicin, basically there is no calcium at all. So it's basically killing the cardiomyocytes. For the Herceptin, we get a lot of arrhythmia, which is, this one of the side effects of the Herceptin. But with the sunitinib, we get actually a calcium overload in the cardiomyocytes, and the tissue is becoming really not moving anymore. So then we looked into the transcriptomics after that. So whether we are going to see the actual phenotypes of these drugs in the heart tissue or not. So we started with the doxorubicin, and when we did transcriptomics on them, we found about 260 genes that are downregulated and 400 genes that are upregulated. And if you look to what's really upregulated or downregulated, so the upregulated genes in the doxorubicin are the oxidation reduction and inflammatory response. And that's what you get basically from doxorubicin because it's oxidizing agent, so it's basically inducing oxidation. And eventually, you're putting down all the heart genes, so basically you are killing the cardiomyocytes and most of the transcription as well because it's targeting the DNA. So what about the Herceptin, so the HER2 or the trastuzumab? So we get as well a few hundred genes up and down, but interestingly, the Herceptin is known to target the contractility of the cardiomyocytes. And you can see that most of the Herceptin effect is showing on the skeletal muscle and the cardiac muscle genes that are downregulated. So it's basically putting down the cardiac gene expression, as well as it's actually increasing some proliferation. Probably this is coming from the fibroblast mostly, but we didn't really distinguish between the fibroblast and the cardiomyocytes in this setting anyway because this is bulk RNA-seq. So what about the sunitinib? So the sunitinib is known to affect the microvasculature mainly and the calcium transients. And what's really cool is this is really showing up in the RNA-seq. So we can see genes that are involved in angiogenesis here and blood vessel formation. And as well as there's inflammatory response that's been upregulated as well. So what are the remaining challenges that we are working on or we have basically tackled? So one of the major challenges is the availability and accessibility of fresh human heart tissue. And basically, Anabio's is addressing one of these major issues. And I think that's why they were really the best home for this technology, I think. And the other thing is, in this system that we have established, we are not really emulating the full cardiac cycle. So we are just electrically stimulating them, right? But the cardiac cycle has got 4 phases. There's systole and then end systolic relaxation, and then end systole, and then diastole, and then end diastole. So there are 4 phases that the tissue has to go through. And this probably could be a reason that we only keep them for 6 days. And there's as well further optimization need to be done for the nutrients and the culture media to make them even go longer than the 6 days. And there's another thing that if we do, I think this will take this technology to a next phase, which is continuous monitoring of these heart slice functionality within the culture. So if we can monitor, so all of the assays that we do, we treat the heart slice in culture, and then we take them out to do the assay. But if we can monitor, for example, the contractility or the electrophysiology or do metabolic assessment while they are still in the culture with the compound, I think this will be really something, it will take it to a different level, right? So this probably will be the end of the marathon, I think. we've worked, actually, I think we worked out a few things recently. We just published this paper 3 months ago in September, so you can go through this paper if you want to have more details, but I'm just going to summarize what we did in this work. So we tried to emulate the mechanics of the cardiac cycle. So in our previous version, the heart slices are sitting in the dish, on supports, and we just electrically stimulate them. They are not moving up and down like they do in the heart. They are not stretching. There's really nothing in there. What's the cardiac cycle? So the cardiac cycle, there are 4 phases. There's the isovolumetric contraction, ejection, isovolumetric relaxation, and then filling. And to emulate this, we have bioengineered a device where we have, I'll just describe from the bottom here. So we have a pneumatic driver that's connected to an air chamber here. And then there's 6 holes on this air chamber, and this is separated by a silicone membrane. And this silicone membrane is connected to another chamber where this column is covered by media, and then the heart slice is laying on the top of this cover on a ring. And then there is a cover for the device, which has got graphite electrodes around each of the slice. So we have 6 slices. And these graphite electrodes are providing electrical stimulation. And this is the device when it gets assembled. So you have an air chamber here, and you have a silicone membrane that separates the media and the slice, and you have the graphite electrodes. And we can pump in and out air from this chamber, and this will move the silicone membrane up and down to make the heart slice moving up and down, to make them stretching like what they do in the heart, okay? And to show you this, these are the pressure volume loops that we get, or how that changes in the pressure in the device. And we have been able to emulate all the 4 phases of the cardiac cycle with almost the exact time. So these are the target times that we targeted, and these are the time that we achieved. So we are a bit 30 millisecond deficient on this one, 10 millisecond deficient on this one, but everything else is almost to the target. And this is the device in the culture, so we can operate 4 devices at the same time. So this is, each one has got 6. And we have 2 pneumatic drivers, so we can operate 8 at a time. So you can basically multiply it, the number of pneumatic drivers that you have and the CPS system that you have. And I think display here, and this one. So, and we apply the electrical stimulation at the end of the diastolic phase. It's about 80 milliseconds before the systole starts. And this is a feedback loop between the pneumatic driver and the electrical stimulator. So there's a feedback loop that basically triggers the electrical stimulation. And you can see here with these, we can basically, I don't know if you can see it, the hearts are moving up and down every second, and the electrical stimulation is being fired at the same time. So, okay, and these are just to show you that this is the chamber pressure, how the changes in the chamber pressure is going, and how the changes in the fluid chamber underneath the slice. And this is the slice movement, so everything basically laying in correctly. And this is how much percentage stretch that we are doing for the heart slice. So we are doing about 25% stretch, which is it's the same like the fractional shortening that you get in the heart. And with the electrical stimulation, so we have developed this technology. This is actually a cool technology. You can get more details about it in the paper. But we are videoing the slice in culture with a high frame rate camera, where we can basically see how the strain is around within the slice on all of the regions of the slice. And at the time of the electrical stimulation, with the stimulated slice, we can get more stretch, more strain basically which is due to the electrical stimulation compared without the electrical stimulation. So this is the same slice with and without the electrical stimulation. And with this technology, we've been able to keep them actually going very well, at least structurally and transcriptionally, as well as functionally, for 12 days. So as you can see here, this one. So with this new system, we've been able to keep the structure of the troponin as well as the connexin 43, as you can see here, as well as transcriptionally, we got about 30 genes are differentially regulated from the 30,000 genes that have been sequenced. And functionally, they are actually functional you can see the calcium transients as well, and they can beat as usual. So can we do some induction of disease model on this? So we've been able to, so you can do a few things on this system, actually. So we've been able to overstretch these tissues to induce cardiac hypertrophy or to induce pathological hypertrophy in these tissues. So instead of the 25% stretch, we've been able to stretch them by about 35%, so that's 10% more stretch. So that's like pressure overload in the heart. And with this overstretching, we've been able to at least induce BMP release in these tissues, as well as getting increase in the cell size of these. So it's a bit of increase in cardiac hypertrophy. So I think to conclude this, I think we've been able to emulate the mechanical stimulation during the cardiac cycle in culture and prolong the lifespan of the heart slices for about 12 days. And then with this culture system, with this new culture system, I think we can emulate different physiological and pathophysiological hemodynamics in culture. You could do other things. You can do ischemia, you could do other stuff, but we haven't really done too much yet. We just published this paper and we are working on other stuff. What's really the next stage of this? What we are working on now is to, you know, you guys heard about the brain-heart axis and the lung and the liver-heart axis and the effect from the kidney to the heart as well, and so on. So now we are trying to do a perfusion system where we can culture slices from other organs, and whatever being secreted from these other organs could be circulating on the heart slices and then go back to the other organs. So this will be like a body on a chip idea. I don't have too much on this yet. This is still work in progress in the lab, but I think it's really cool. But hopefully within a year or two, we can show you some more on this. At the end of the talk, I would like to thank my group, which are really doing an awesome job on this. We get human hearts at midnight, and they have to stay all night to process these. So they've been working really 24/7 on this, and on other stuff as well. This is not the only thing that we are doing. As well as the NIH, the DoD, and Merck for funding, as well as our collaborator around the country, and Anna Byers as well for their collaboration. Thank you so much.
Thank you, Tamer, for a great insight into your research. Any questions?
Thank you for the excellent talk. Excellent technologies and so on. And I will say, I think you'll be intrigued by some of my presentation later this morning on complementary approaches in the lung. But that's for later. Yeah, I do have a pointed question. In your stretch devices, you talk about strain. What does that really mean? Radial strain, normal strain, axial strain? And I see the profile is pretty heterogeneous. And that to me is confusing when you talk about a stretch device.
So I think the difference, so I will explain to you, I'm sorry, I just skipped on this. So each number of these, so we have divided the heart slice into 10 contours across so they are circular contours. One is the center to 10 is the outside support. And you can see most of the strain is happening in the middle of the slice because the other two, the center as well as the supports are stable because the hard slice is stuck and the center doesn't really have too much strain. All the strain is coming from the other contours that are in the middle. So, and we have, this was done by the bioengineers. It's been explained very well in the paper. So if you are, you can go back to the paper and they've used the model of a cone thing. That's what I remember. So it's a cone simulation model to basically get this strain. But actually, it's the whole strain in the contour. So it's 10 contours around the slice. And you can see all of the electrical stimulation that you can see the differences in is between 3 to 8, right? So this is basically the thing that's away from the solid support, and it's not the middle as well. So the middle doesn't really have too much strain in there.
Thank you.
Great talk. I was just wondering, in the first part of your talk, you were referring to some comparison studies looking at toxicity between these cardiac slices and IPS-derived samples. So I'm wondering what your benchmark is in regard to the IPS cell biology. There is certainly a lot of effort, and you're working with James Hudson, for example, in regard to the bioengineered heart tissues. And James would certainly say that the IPS-derived engineered tissues are very close to an adult or really progressing significantly toward that in terms of efficacy studies for toxicity. So, what is your benchmark in regard to comparing the slices to IPS-derived matured, at least pushing in that direction?
Yeah, I fully agree. So what we did with the IPS was monolayer on the cardiac site. And as I said, there's another 37 sites. Some of these sites are doing EHTs. So all of the data now are with a statistician to really have a consortium-wide analysis for these data. I just got a permission from them to show my data. And they allowed me, so that's why. But I think there will be a publication around the whole consortium. So this is basically version 2 of the SIPA trial, and this will be. I don't know about the other data yet, so I didn't get the other data. So yeah, I just give the data back. All right.
Tamer, very nice talk. I have a technical question regarding the dynamics over the 12 days. So I want to just understand this correctly. Of course, you are putting in air, which is pushing up pressure on the liquid on the chamber below the slice, and that pushes up. And of course, you're doing this repetitively. Now, conceptually, I was thinking about this. So if the tissue itself is somewhat permeable to fluid, some of that fluid will get across into the other chamber, and that will drive the pressure on that side higher. So you would have to apply more and more to keep it the same deflection stable, unless, of course, you can relieve the pressure on the other side. Does that happen, and is this stable over 12 days?
So yeah, so basically, the first version, so this is, I'm showing just the success, not the failure. And it was 90% failure and 10% success. Okay, so the first iteration was to pressurize the whole chamber from up and down. Okay, and this failed because really the heart is contracting from one side and you don't have the pressure from the other side. You have to leave it. And as you said, if you build up pressure on the top, you can't really, you get more pressure on it building up, right? But as you can see here, so the level of fluid is stable, okay? And I agree with you, so the tissue could be semi-permeable, but this every second, basically we are going for 72 beats per second, right? So we are pushing up and down every second. So whatever goes up, goes down. So there's really no build-up, because whatever perfuse up, it will perfuse down again. And we have an air and it's not sealed. So there is really no pressure build-up from the top. And this was the trick. I think we were failing for a year because we were pressurizing it from both sides. But I think the success was when we unpressurize it from the top. But it's a great point. And it's actually, we run through it, so yeah.
Thank you, Tamer. You're doing cool stuff. My question is about the different sensitivity to toxins that you see in your slices compared to the iPS cardiomyocytes. And I'm curious about your thoughts on that, because there's a lot of differences between the two systems, right? I mean, one option is the maturity, but also you have all the other cells in the slices that could help mediate some of those responses. So I just wonder, what are your thoughts at the moment? Like, why is one system much more sensitive than the other?
Yeah, so I think this is a great question. I think two things. One is the maturity, and the other thing is having global structure of the whole myocardium. Because sunitinib, for example, is affecting microvasculature. So the IPS doesn't have microvasculature, right? And basically, we haven't detected anything structurally on the heart slices, because we are looking into the cardiomyocytes as well. But we still, you know what we detected when we did the RNA-seq, because it's affecting the gene expression in the microvasculature, so we can detect it when we did the RNA-seq. So you have to have, I think you have to have a full battery of assays and biomarkers, because you have to assess everything, plus the additional tissue integrity that you have, and the crosstalk between. I think we just had this amazing talk from Ted, where he showed the spatial transcriptomics and how things are interacting together. And basically, I think this interaction is never been on even any microtissues. All of the iPS-derived microtissues, they just dump all the cells together and they form a microtissue. So there is really not an organized way of doing it, at least.
On the other hand, though, I mean, it probably will be cheaper and you will have more scale to do it with the iPS cardiomyocytes. So I like your idea that this would be a secondary.
Yeah.
Because if something is already toxic in the iPS-derived cardiomyocytes, it's just going to be toxic anyway.
I would tell you one comment from the FDA director of drug discovery. And he told me, this was in 2020 when I talked to him after our testing the 3 compounds. He told me, you really want me to make this as an essay and tell drug companies to do that? I said, yeah, why not? And he said, okay, so then you're going to be rich, and then the drug companies are going to kill me, because this is not going to happen, and they're not going to do it. I said, okay, so what shall I do? He said, convince them to do it. So that's why, that's why I've been doing this marathon eventually, and trying to, to spread this technology as much as we can. And I would say it's a secondary. I think it's better to be secondary assay, because there's really no need to screen 1 million compound on heart slices. You can just, your final target, your final candidate that you want to go forward with, test it on heart slices, see how it works, see whether it's toxic or not. If it is not, that's fine. You can go ahead and do the clinical trial. If it's toxic, so stop it. It's much better than the dog assay. So all of the drug companies now are doing dog assay, basically. This is the highest standard that you can do before you move to the clinical trial, right? So yeah.
It's also excellent for heart failure drug discovery.
Oh, yeah,
Nanobios,
this
we
is
know
a-
that it's difficult to make cardiomyocytes from heart failure donors'
Yeah.
hearts. So it's quite enabling for that.
Yeah.
But thank you very much, Tamer. Appreciate your great presentation.